Good morning, students. Now coming to translation. This is the second phase of protein synthesis. The first phase of protein synthesis is called transcription that takes place in nucleus. Here, genetic information is copied onto mRNA. But the second phase, this is called translation. It takes place in cytosol. And this is the second phase of protein synthesis. So translation means formation of proteins. Okay, formation of proteins from mRNA. This is called translation. At the same time, this translation is also called formation of a polypeptide chain as per the codons of mRNA. That is called translation. Okay. So here, during the time of this translation, as per the codons of mRNA, based on the sequence of the codons of mRNA, amino acids they are linked one to each other, leading to the formation of a polypeptide chain. A polypeptide chain. Okay. Based on the sequence of codons present in mRNA, amino acids are linked one to another by peptide bonds, leading to the formation of a polypeptide chain. So, translation process it is dictated by DNA and represented by mRNA. As per the codons of mRNA, many amino acids are linked one to another by peptide bonds to form a polypeptide chain. Such formation of a polypeptide chain as per the codons of mRNA, that is called translation. Students, 20 types of amino acids. Okay, 20 types of amino acids they are present inactively in cytosol. Inactively, 20 types of amino acids are inactively present in cytosol. Okay, now during the time of this polypeptide chain formation, every amino acid it consumes one molecule of energy, adenosine triphosphate, in the presence of enzyme, one enzyme, that is amino acyl synthetase, and in the presence of tRNA. Every amino acid consumes one ATP molecule in the presence of amino acyl synthetase enzyme. Now it becomes activated and then it is attached with the CCA end of tRNA. Already know the tRNA structure in previous class. There are total 5 loops. CCA end is one of the loop. Now this activated amino acid it attached with the CCA end of tRNA like this. For example, this is activated amino acid and this is tRNA, one loop, and this is another loop, anticodon loop, and this is tRNA. So, amino acid it is activated in the presence of enzyme amino acid synthetase and by using one molecule of ATP. And then it is attached with the CCA end of tRNA. Now, this tRNA is called the tRNA with the activated amino acid which is attached with this CCA end. Such tRNA, this is called amino acyl tRNA. This is amino acyl tRNA. Okay. This formation of amino acyl tRNA, this process, which process? Activation of amino acid and attachment of amino acid to CCA end of tRNA. This is called charging of tRNA. This is called charging of tRNA. This is also called amino acylation of tRNA. Amino acylation of tRNA. This is most important. Amino acylation of tRNA or charging of tRNA. Okay, this is charging of tRNA. Students, you know, in case of prokaryotes, the tRNA that transfer the first amino acid methionine, formyl methionine, that is called formyl methionine tRNA. Formyl methionine tRNA is also called amino acyl tRNA. Initiation tRNA, initiation tRNA, initiator tRNA, like that. Okay, now coming to another point, ribosome. Okay, ribosome, this is a responsible major cell organelle for the protein synthesis. Protein synthesis takes place in ribosome. This is also called factories of proteins. Ribosome is called factory, protein factory. This ribosome is represented by ribosomal RNA and 80 types of proteins which are present inactively. 80 types of proteins and RNA, they are present inside this ribosome. Similarly, this ribosome is also represented by smaller subunits and larger subunits. Smaller and larger subunits. There are two units, large and small. For example, in case of 70S ribosome, smaller subunit is 30S and larger subunit is 50S. Like that, every ribosome is represented by two subunits, smaller subunit and larger subunit. Similarly, this ribosome is also represented by, okay, this is also represented by larger subunit. Larger subunit, it also contains A site, acylation, amino acylation site, A site, and peptidyl site, P site. There are two sites. Okay, peptidyl transferase enzyme, which is responsible for peptide bond formation, is also available. A site and P site. There are two sites. First, the smaller subunit. Okay, smaller subunit, it attached with mRNA. First, it is attached with mRNA, like this. For example, this is mRNA, 5' region and 3' region. This is mRNA and UTR regions, okay, and this is smaller subunit and this is larger subunit. This larger subunit is represented by two sites. One is P site and other one is A site. There are two sites. Now the smaller subunit is attached with mRNA. This is AUG codon. For example, second codon A, third codon G, and like this last codon UAA. This is mRNA. For example, AUG codon is present in P site and triple A codon is present in A site, and triple G codon present outside to the largest subunit of ribosome. Okay, students. Now the tRNA AUG codon, it recognizes methionine. Now methionine is transferred by a specific tRNA. Now this tRNA, it recognizes methionine and the presence of one molecule of ATP in the presence of enzyme. It is charged with tRNA responsible for AUG codon is charged with methionine amino acid. Now that is considered as formyl methionine tRNA. Now this formyl methionine tRNA, it entered into the P site of larger subunit. This is methionine large subunit. So the relevant tRNA, it recognizes the amino acid methionine and then it becomes charged and it becomes amino acyl tRNA and then it transfers the methionine into the P site of large subunit. So anticodons of tRNA, they are complementary with codons of mRNA. What letters are present in anticodon of this tRNA, that is initiating tRNA that transfer formyl methionine amino acid? A is complementary with U and U is complementary with A and G is complementary with C. Okay, in this way this tRNA is attached with AUG codon at P site. Now this complex is called initiation complex. This is called initiation complex. At the same time, another tRNA which is responsible for second codon also recognize the second amino acid. For example, it will be A. It recognizes lysine amino acid. Lysine amino acid is also activated by using one ATP molecule. Then it is attached with the relevant tRNA. Now this tRNA, it also transfer the second amino acid lysine and it is sent into the larger subunit. Now the anticodons of this tRNA, U. Anticodons of tRNA is U. So now this anticodons of this tRNA is complementary with codons of mRNA. So A complementary with U, like that. Now A site and P site, they are filled with two different types of amino acids Now a peptide bond is formed between carboxyl group of first amino acid and amino group of the second amino acid by releasing one water molecule, leading to the formation of a peptide bond. This CONH is considered as a peptide bond. This is called peptide bond. So this methionine, it is shifted into the A site and then it is attached with the lysine amino acid by one peptide bond. So this tRNA, it becomes empty. This tRNA becomes empty. Now this methionine first amino acid is shifted into A site and attached with the second amino acid lysine by one peptide bond. And this peptide bond is formed by the process of dehydration. Now this tRNA, it becomes empty and then it leaves the largest subunit of ribosome. Its work is completed. It leaves the largest subunit of ribosome. Thus, a peptide bond is formed between the carboxyl group of first amino acid and the amino group of the next amino acid. So that this amino acid is shifted into A site and is attached with the second amino acid. Now, this ribosome, it migrates to the next codon. The movement of ribosome along the mRNA from 5' region to 3' region in one codon length, such a movement of ribosome is called translocation. This is called translocation. This translocation, it also requires one molecule of GTP. And bond formation, it also required one molecule of GTP. For activation of amino acid, every amino acid it requires one ATP. And for bond formation and translocation of the ribosome, every amino acid it requires two GTPs. But first amino acid methionine, it requires only 1 GTP because translocation is not required for methionine. Why? Methionine is directly entered to the P site. So, except methionine and all amino acids, they require 2 GTP, but methionine it requires only 1 GTP. So, translocation takes place. Now, because of this translocation, AUC codon it leaves the largest subunit, and AAA codon it migrates to P site, and third codon GG it migrates to A site. Now, another tRNA it recognizes glycine and also brings this glycine into A site and bond formation. So, incoming of charged tRNAs into A site and bond formation, migration of ribosome, and all these three events takes place continuously until one of the ending codon is entered into the ribosome. One by one, it decodes the genetic information of mRNA and continued up to until one stop codon is entered into the A site. Once the stop codon is entered into the A site, these stop codons they do not recognize any amino acid. They have no amino acids. They have no tRNAs. So charged tRNAs into the A site will be switched off. Now releasing factors they recognize this UAA stop codons, and finally this process it terminates by terminating codons. This is the concept. Now coming to some objective points. Okay, students, for example, one polypeptide chain is formed by 25 amino acids. One polypeptide chain is formed by total 25 amino acids. Okay, what is the total number of codons present in mRNA? 25 amino acids, they require 25 sensible codons. To terminate the process, one additional AUU codon, any stop codon is required. 25 plus 1, that is 26 codons are required. Okay, for example, number of amino acids, if it is considered as n, number of total codons n plus 1. This is n plus 1. N means total number of amino acids. Every amino acid is recognized by one sensible codon. To terminate the process, one additional any stop codon is also required. Including the stop codon, total required codons present in mRNA, that is 26. Now coming to the point, total number of sensible codons. Always sensible codons is equal to the number of amino acids, equal to n. Okay, and what about the total number of n times nucleotides present in mRNA? That is 26 total codons into 3, that is 78. Okay, every codon consists of 3 nucleotides. There are total 26 codons including the stop codon. 26 into 3, that is equal to 78 nucleotides are present in mRNA. At the same time, what about the total number of n types present in a template that provides the information for mRNA is also 78. And total number of n types present in such a segment of DNA. Actually, mRNA is formed from one segment of DNA. mRNA is formed from one segment of one strand of DNA. One strand is represented by 78 and two strands 78 into 2. 78 into 2. Okay, and what about the required number of ATP? Required ATP, always number of required ATP is equal to the number of amino acids. This is also 25 is equal to n. Now coming to our point, what about the required number of GTP? Every amino acid it requires 2 GTP except methionine. Methionine it requires only 1 GTP. So 49, that is 2n minus 1. 2n minus 1. And what about the total number of peptide bonds? Peptide bonds are also 24. 24 peptide bonds, n minus 1. And what about the required number of tRNAs participated? tRNAs participated always equal to number 25 equal to n. And what about the translocations complete in the ribosome? Translocations always equal to n minus 1. Okay, this is the information that is most important for objective purpose.